Good afternoon, everyone, and a huge welcome to the much anticipated Asian Science Camp 2025. My name is Pasit Pakawat Panurat, and I'm thrilled to be your MC for this exciting event. Over the past few days, you, our brilliant young scientists, have been hard at work developing your poster projects. We've seen incredible dedication encouragement. Now it's time to share your brilliance on stage. Today we will hear from 10 groups who have been selected from the poster session. They will be pitching their innovative ideas centered around three crucial themes: sustainability, innovation and technology, and fundamental and creative concepts. So let's get ready for an afternoon of inspiring ideas and groundbreaking concepts. Are you all ready to hear our pitchers? Okay, excellent. Now, to ensure fairness for every group, let's go over how the timing for the pitches will work. Each team has a strict 3-minute time limit for their presentation. To help our presenters manage time effectively, we will be using the following signals. First, you hear one gentle ring of a bell. Okay, one gentle ring of a bell at 2 minutes and 30 seconds. When you hear this, it is your cue to begin your concluding remarks. And next, you will hear a beep sound. A beep sound. At 3-minute mark. This signals the official end of your presentation time. However, should you continue with your presentation, you're gonna hear something like this. Okay, all right, to signal that you have to really stop, okay? We kindly ask all presenters to respect these signals to ensure every group gets their full and fair amount of time. Please also note there will be no Q&A session after each pitch. All right, the rules are clear, the energy is high, and our presenters are ready. Let's begin now. Let's welcome our first group to the stage. We're talking about Group 7. Group 7, are you ready? Please be on stage, which we will be presenting on the theme of sustainability. Group 7, welcome. You can stand in the center of the stage. Okay. All right, are you ready, Group 7?
Yeah.
Okay, if you're ready, your 3 minutes start now.
So good evening, everyone. Today I'll be presenting our project on the topic compact sustainable tree technology. So as we all know, since population is increasing drastically and the amount of land area that is available for planting trees is decreasing, so we've come up with this idea of having a compact tree which can be installed on houses and buildings, wherein you not require the amount of space that a tree takes, and still do the same processes that a tree does, that is photosynthesis, and also energy generation. So the model that we are planning converts carbon dioxide to oxygen, thereby cleaning air, and at the same time, it also produces energy by photosynthesis. The brief structure of the model is as such. First of all, it has a protective layer, which is made up of recycled plastic, followed by a photosynthetic layer, which contains a dye called spaudarine dye, and a modified version of that, which is more conjugated in order to capture sunlight, a broader spectrum of the sunlight, and it also has cyanobacteria. So the dye captures sunlight and transfers it to cyanobacteria, which performs photosynthesis and gives out oxygen, and the energy that is produced is converted to electricity. This electricity is then transferred through ion migration channels, which is the third layer of the device, and can be used for various applications. Now, my team member will be explaining the mechanism.
Okay, so the first two questions we're facing about is very important. The first one is the energy transfer efficiency. We know that the energy transfer in here is very distance sensitive, so that we need to make sure that the distance between those organic dye molecules are small enough to ensure a efficient transfer. And to solve that, we applied a technique called spy catcher and spy tag. We basically form a covalence bonding between those organic synthetic dye, and the distance between them get extremely small so that we can promote a high efficiency transfer. And the next problem is that we know that once the ATP molecules with high energy electron are produced within the photosystem 1, we cannot retain them in the cell for a long time because that's unstable. So we applied a CRISPR technique here to edit a gene that blocks cyclic electrical flow so that we have a surplus for electrons. Yeah.
Now, once we get the electron, we want to bring this electron and convert it into electrical energy. The method that we use is we use a molecule called Nd, which goes into the cyanobacteria and attaches to the electron. It goes out, and then it distributes this electron into the CNT anode, which creates electricity. There are some problems, such as the efficiency. It's better than a normal tree, but not as efficient as a solar cell. Thank you.
Wow. Perfect timing. Thank you, Group 7, for that fantastic presentation. Let's have a big round of applause once more. What a great start. Now let's move on to our next presentation. Our second group, which is Group 37. Group 37 will be focusing on the theme of sustainability. Group 37. Go to the stage. And next group will do the same thing. 3 ตัว Three? Yes. Yes. Over there on stage. Okay, let's give a big hand to group 37. Group 37, you ready, right? You ready? And looks like you're ready now. So you have 3 minutes and your time begins now.
Dad, I want to see the fish.
Darling, you can't because it's already extincted.
Humans have been unintentionally affecting the marine biology around us through pollution and climate change. For example, a statistic recorded in 2021, there are 17 million tons of plastic in the ocean today, and this number is expected to double or triple by 2040. Before we get to the solutions, we will be explaining some of the extinct species, some of the species that are endangered, and the effects that it can have when extinct.
Okay, so first, endangered species, as you can see, there's like 10 of those. So basically, the first one that appears is the name. The second one is the type of animal. The third one is how many animals are there in this world. And then the fourth one is why they went extinct. And as you can see, it's also accompanied by the pictures.
So surely, as these species are part of our ecosystem, so they will have effect on climate, they will affect on human, and they will surely affect on the ecosystem. So the first and foremost will be like there will be increase in the temperature, and the sequence of that there will be polar regions, and as a result of it, there will be increase in the sea level, and the islands will get. And the second is human societies. The human societies that are living along the coastal lines will as they depend on these species for food, so we'll go to the city area, we get a lot of tourism. And the third one is like the collapse of the ecosystem. As all of us are the part of ecosystem, when these species will get extinct, what will happen? We will get the loss of biodiversity, and this will lead to the acidification of the oceans, and there will be damage to coral reefs too.
Now that you guys know some of the effects that this can have, we will be starting to explain the solutions. Solution number 1 will be presented by Sahav.
So the first solution and the foremost solution is most of the damage to the ocean species is by the plastic bags. So we can remove them by placing AI deep water cleaning machines that will be integrated with satellites, and they will mark the hotspots that are rich in that have much of the plastic bag. So they will suck them and they will clean the water.
The second solution we suggest is implanting computerized chips. We will be implanting computerized chips that will not be affecting the lifestyle, and it will be tracking and monitoring the location and vitals. And if these vitals go abnormal and out of range, then it will alert nearby marine biologists to try to save these animals from dying.
And lastly, with the genomic library, so basically you just collect the DNA of this endangered animal and keep it for the future. Imagine if they all died, what are we gonna do? We might use the future technologies and clone it by using the DNA that we keep in our lambda phage. And this is all about our poster, Turn the Tides for Ocean Life. Thank you for your listening.
Right on time. Thank you. Wonderful job, Group 37. Thank you for sharing your project with us. And now, for our third pitch of this afternoon, which is Group number 9. Group number 9, where are you? Please be on stage. Okay, Group 9, you have 3 minutes and your time starts now.
Hello, ladies and gentlemen. 13.7 million global deaths yearly. Infectious diseases. So, ladies and gentlemen, this is the load of the infectious diseases in the world. So, basically, we are having a lot of infections, infectious diseases, the viruses like HIV, polio virus, Congo virus, and the list continues. Lately, 2 years ago, 5 years ago, it's been a lot of time, COVID virus came in the world, and we required a lot of months to make a vaccination against it. So me and my team have presented the idea of making a microchip. This microchip will work as a biological firewall, and it will be made of biodegradable material, graphene. So now, this chip will basically use the technology of CRISPR-Cas system. The CRISPR-Cas system will be able to detect the virus as soon as it enters inside the human body. We will implant this chip inside the subcutaneous deltoid muscle. So, number one, this chip will identify the type of virus through biosensing, censoring. And number two, when it will be able to identify the sort of virus that has come inside the body, what will it do? It will trigger the neurotransmitters like cytokines to trigger the immune system. Hey, let's fight the virus. So basically, if we talk about some of the biology, the viruses take a few days to infect the body, like 3 or 4 days. So our chip, it will what will it do? That it will detect the virus on the very first day, and by activating the immune system, it will kill the virus on the very first day before it is even infectious. So basically, we have presented this idea as an alternative of the vaccinations. Billions of dollars are spent on making of the vaccinations by the pharmaceutical companies, and there are the logistic economics too involved in it. So we can save all those money and spend it on the microchips. What will be this benefit? The quality of life of the people will improve, and eventually we aim to eradicate the polio virus from the world, Congo virus, HIV, and most importantly, we will be able to combat against all the new epidemics that are gonna come. Like there was a talk that every 5 years we're gonna have a new epidemic, so we will be able to combat against them. Thank you so much.
Thank you, Group number 9, for a nice presentation. We have so many interesting pitches this afternoon, and we have many more to go. Now let's welcome our fourth group to the stage, which is Group number 5. Group number 5. This group will be presenting on the theme of innovation and technology. Welcome group number 5. Please give a warm welcome to group number 5 once more. And your 3 minutes start now.
Have everyone know about a gum disease? Okay, so a gum disease is basically an infection of your gums from the bacteria. And in the early stage, actually, this gum disease it didn't even show any symptoms, so it's invisible. But once it's developed into the last stage, it will show some symptoms such as the bleeding and also some of the swollen gums. So we want to test this gum disease in an early stage so that we don't have to be too late for us to cure it or to treat it. And here is how our test kits work. So you can see that basically it has 2 parts. So the first part is inside of your mouth and the second part is outside of your mouth. So look at the first part first. So the first part, you have to know about the interleukin. So interleukin is something that our body will produce when we have a gum disease. And after that, this interleukin will flow inside of your oral cavity. And after that, here is come to our patch. So our patch in the first layer, it's the MIP or the molecular imprinted polymer. So this polymer can capture some specific molecules. So in this case, it is the interleukin, as I mentioned earlier. And after it's captured together, it will bind, and this thing will make the MIP become swollen or become bigger or broaden. And after that, this MIP, as it's become bigger, it will push down the layer beneath it, which is the TENG or triboelectric nanogenerator. So this generator will produce energy when it has some physical force that press onto it. So basically, it's turned the physical force to the electric force. And after that, we will send the amount of the energy that TENG produced to the second part, which is the part outside of your mouth. So you can see that there are some mechanical system that will translate the amount of the electricity into the data that will send into your phone and to the application for the patients to see the result. So from this graph, you can see that in normal people, as I mentioned, that they will not produce interleukin or produce in a very stable stage. So you can see that this graph, which is the graph of the energy that TENG made, will be quite stable. While people who have gum disease, their graph will be quite rising because their body producing the interleukin, so that the TENG again here will produce the energy and make the graph become rising. And here's come to an end of my presentation. So lastly, I want to say that strong smile begin with healthy gum. Get checked early and be healthy. Thank you.
Thank you, group number 5. The time is too short, isn't it? So now let's move on to our next presentation. Our next group, which is group number 33. Group number 33. We'll be focusing on the theme of innovation and technology. Please welcome group number 33. Group number 3, if you're ready, your 3 minutes start now.
Hello, everybody. Hello. Yeah, so when was the last time that you actually had an 8 hours of good sleep? Probably a long time ago, right? We all know the importance of sleep, and when you are lack of sleep, that it can do serious damage to your body, both physically and both mentally. That's why we have come up with the design for the Super Ultra Deluxe Super Pod, Sleeping Pod, in order to help you get the quality of a 10-hour sleep in only 2 hours. And it will be a fully customizable device, so you can just go there, get a sleep, get out fresh. And how exactly does it work? Well, my friends here are here to tell you.
So the first feature is the Elamine. It's an ambulatory wearable electroencephalography-based neuromodulation system that transmits low-frequency waves that stimulates the brain to produce theta waves. And theta waves is what induces you to go into deep sleep. Secondly, we have the red and blue light light system. Red light stimulates the production of melatonin to relax the body and induce sleep, while blue light acts as an alarm clock to rule users back awake and remove melatonin from the body. Next, we got binaural beats, which is when two different frequencies are played into each ear, increasing low frequency brainwaves and decreasing high frequency brainwaves. This induces drowsiness and reduces cortical hyperarousal.
After that, we have the scent dispenser. Oh, I have a perfume in my hand. Okay, so what does it do? Studies have shown that jasmine and lavender scent have a great effect on your sleep. And it's been proved now that these scents slow your heart rate, they reduce your sympathetic nerve activity and increase your parasympathetic activity of the nervous system and relaxing the overall body. After that, we have a temperature control unit in the pod that will control the temperature of your pod according to your need. After that, we have a health monitoring system. Oh, I really want to talk about that. AI health monitoring system, it will constantly read your vitals, your brain waves, your heart waves, the oxygen concentration in your body, your motion of your body, and it will constantly change the conditions of the pod according to your vitals. And one other thing it do, for example, God forbid, if you had a heart attack when you're lying in bed, so this system will contact the doctors, contact the authorities, so you can just get the right treatment on the right time. We have some limitations. Obviously, it was going to be expensive. The second is Elon Musk. How's Elon Musk a limitation? So it's exactly, if the people on Earth say we don't want to sleep in a pod, we want to sleep in a room, so then we are going to use these pods in the spaceship that Elon Musk is building for Mars. And the third is, we don't know the side effects yet. We can only know that by clinical trials. Thank you very much.
Good job, Group 33. Thank you for sharing your project with us. And now, onward to our sixth pitch of this afternoon, which is Group 44. Group 44. This project is centered on the theme of innovation and technology as well. Please put your hands together for Group 44. Group 44, when you're ready, the time can start now.
Hello, ladies and gentlemen. My name is Indy, and today I'll present you Hera. Well, in our world, we are following the trend of the AI trend, aging trend, and also the trend of the natural disaster. That's why I try to come up with the innovations that create in a systematic in civil engineering. Well, the first one, we start out with the satellites. It's called a Sky Sentinel microsatellite. Sky Sentinel means run by the power of AI while still have the ground stationary that still receives the signal. While swapping to the microchips, it reduces the production cost, which allow the governments to buy these satellites in the very large amounts and create a more effective results in the kind of natural disaster defector. And the microchips that I insert, it has ASIC, which stands for Application Specific Circuits, NPU, which stands for Neural Processing Units, and the Quantum Encryptic. These microchips are responsible for generating pictures, stitching the small pictures from the stationery, and also the Quantum Encryptic that ensures the stabilized signals and also a clear picture to send down to the neural disaster web, which is the AI powerhouse of data sets that were given by the governments and actually store a very large amount of data about the elders, about how some people will actually eventually act in some kind of certain circumstances in a natural disaster. And it was sent into the resilient beacon. The reason why I use resilience is because it acts also as a shelter for people, while the beacon means the light, the guiding lights, which contains a synchrotron panel. The way it was different from a solar panel is because it contains another layer called graphene. Graphenes can accumulate the radiation signals from the synchrotrons. Well, synchrotrons happen from when electron accelerates, emits the light, and that light is called synchrotron radiation. And before we emit that, we're going to diffract that electromagnetic waves into X-ray, and then emit this out, and then define the structural, define how people trapped in the building, and actually define how some people can actually act in some certain circumstance by using the data sets from the neural disaster web. Before sending back into the satellites, and the signal will change into the cosmic ray, which is the muon. And then it generates by creating the pictures from numerous stationaries, stitch the picture together, wrap it up, and then send down to the government and personal phones in order for the people to actually eventually escape and also have, have a safety in that kind of situation. Thank you.
Thank you, Group 44. What a nice presentation. Let's now welcome our next group, which is Group 47, to the stage. This group will be presenting on the theme of, once again, innovation and technology. Please give a warm welcome to group number 47. Group 47, your 3 minutes start now.
Hello everyone, we are Group 47 and the title for our subject is Space Waste Hunter. Our problem we want to solve is that there are tons of space debris outside of our atmosphere and we really want to clean it up. So we have two goals for our subject. The first one is to create a spider-like spaceship, looks like this one, and with arms, and also it will eject webs to be able to collect the space debris. And the second goal we want to do it is we want to send these space debris to the International Space Station, and also use bioengineering skills to be able to degrade the plastic and metal inside the space debris.
So how AI scan debris? As for the spider-like satellites, its arm has the cameras and sensor for real-time imaging, so the AI can identify the plastic, metal chips, and sensor and shoot out the webs targeted the debris. Inspired by the spider webs, the net webs around debris with magnetic edges. So what happened after collection? So debris is pulled back and stored in the satellite. When full, the satellite docks at the space stations. The material are sent back for recycling or safe disposal.
Okay, for the next part is, for the trash is going to inside the ISS, there is a cabin which includes 3 chambers. Most of the chamber is like firstly and a second is the half part of the cabin. First, we have to look into the chamber number 1, plastic degrading. It's just a hydrogel that consists of enzymes from the bacterium and fungi to dissolve plastic into the acetate and format, which acetate and format is able to transfer from the dissolvable membrane. Next, throughout the chamber number 2, which is where geobacter is consisting in for electron producing state. The geobacter is using acetate and format as a food to produce electron. Electron then transfer and kept inside the capacitor. There we have way down of the first half. The second half is like the chamber number 3, which is metal degrading. To use this, there is a hydrogel consists of producing enzymes to degrade metals into ions and electrons. Both ions and electrons is inside the hydrogel and using the electron from the previous process to make it solidify, and it can be recycling.
And so for wrap up, we want to say that the best innovation for our project is that we try to combine AI technology inside the spider-like spacecraft and also implement bioengineering skills to degrade the metal and plastic and to achieve sustainability. Thank you.
Thank you, Group 47, for sharing your innovative project with us. Now, wonderful. Now, let's move on to our next presentation. Our next group, which is group 11, will be focusing on the theme of innovation and technology once more. Let's give a big hand to group 11. And you have 3 minutes, and your time begins now.
Imagine if we could not just suppress HIV, but delete it. Retroviruses like HIV and HTLV are currently not curable. They don't just replicate in the bloodstream. They integrate their DNA into our genome, hiding in our own cells. Even the best antiviral drugs only silence the virus. They can't remove the hidden DNA, and the moment treatment stops, the virus comes back. To truly cure retroviral infections, we need to go into the genome, find the viral code, and remove it completely. That's what RetroSmasher is here to do. A futuristic therapy that combines AI, CRISPR, and biotechnology into one powerful system to detect, target, and erase retroviruses for good. Here's how it works. It all starts with AI-powered detection. We train deep learning models to scan a patient's genome through blood and tissue. Once we find these cells, we sequence the viral DNA inside them. Since retroviruses like HIV mutate rapidly, we use AI to analyze those sequences and design precise CRISPR guide RNAs for each patient. But the real innovation? These guides are adaptive. As soon as the virus mutates again, the AI detects it in real time, redesigns the guide DNA, RNA, and keeps CRISPR one step ahead. It's like an intelligent, evolving immune system powered by code. Next, we have to deliver CRISPR into every infected cells. We use engineered nanoparticles and viral vectors to carry out CRISPR safely into the body. These carriers are optimized to cross barriers like the blood-brain barrier and to selectively target infected cells. AI helps identify the surface markers of infected cells so we can ensure CRISPR reaches even the most hidden viral locations. Once delivered, CRISPR cuts out the viral DNA and the cell repairs itself, virus-free. But the treatment doesn't just stop here. RetroSmasher keeps working even after CRISPR does its jobs. Our system continuously monitors blood and tissue samples in real time, scanning for any signs of viral rebound or mutation. If it detects a new version of the virus, the AI instantly generates new guide RNAs, and the system deploys another precision attack. This makes RetroSmasher not just a one-time therapy, but a self-updating real-time genome defense system. If we can delete viruses from computers, why not delete viruses from our own DNA? This is RetroSmasher, and together we can the incurable. Thank you.
Thank you, Group 11. What a fantastic project. And now for our 9th pitch of this afternoon, which is Group 41. This project is centered on the theme of fundamental and creative concepts. Group 41, please put your hands together for Group 41. And like other groups, you have 3 minutes, and your time begins now.
Hello, everyone. So our proposed model is targeting immune cold tumors by combining phoroptosis and immune checkpoint inhibitors, ICI, together. So the current challenges in targeting tumor cells is that it avoids apoptosis and has a very dense fibrotic stroma. So this prevents immune cells from reaching the tumor cells. So to overcome this, we're going to induce phoroptosis, a type of cell death in the tumor cell itself, and which it does not rely directly on T-cell access. Instead, the phoroptotic tumor cells releases danger signals, DAMPs, and cytokines that can alert local immune cells that are already within the tumor microenvironment. This also activates bystander T cells in a receptor-independent manner, which boosts the immune response together. But one of the problem is that phoroptotic tumor cells can also release PD-L1 positive exosomes that can silence our tissue resident memory cells. That's when we're going to administer ICIs. These ICIs are going to block the PD-L1, PD-1 axis, thereby reinvigorating the tissue resident memory cells. By inducing phoroptosis and administrating ICIs, we can activate both TREMs and bystander CD8 T cells, and thereby maximizing the immune response.
Now, moving on to optimizing this mechanism, we can start off with the ICI that is being used. Immune checkpoint inhibitors are crucial in order to synergize with ferroptosis. The pharmaceutical pembrolizumab, a PDI drug, shows the highest objective response rate among monotherapeutic drugs, meaning it shrinks tumors the most. Secondly, the time of treatment of synergy is also important, as we recommend the induction of ferroptosis first, and then the administration of ICIs.
For in the future and SDG development, we wish it firstly offer a solution for immune cold tumor, and then minimize systemic side effects by leveraging TRM cells. And the main one, open up new strategy for precise immunotherapies. I wish it can be adapted across multiple cancer types. For our projects, it's on sustainable development goal number 3, that is especially 3.4 and 3.B. Thank you for your attention.
Thank you, Group 41, for a very nice presentation. Now, let's welcome our last group, which is Group 48, to the stage. This group will be presenting on the theme of fundamental and creative concepts. Please give a warm welcome to Group 48. And looks like you're ready, and your 3 minutes start now.
Hi everyone, we are from Group 48, and we are here to present you the OptiCortex. But before we tell you about the product, let me tell you why the world actually needs this product. As of recently, it has been reported that the approximate number of people suffering from visual impairment is 2.2 billion. Even worse, 1 billion of them actually could be prevented. So you must be thinking how it will affect you. It has been reported again that the annual economic loss from visual impairment is approximately $411 billion, an astonishing amount indeed. Thus, we must propose this solution, OptiCortex. Now, Pim will tell everyone about how this device functions.
Okay, for the methodology, you can see a black one here. This is the height-mounted camera system continuously capture real-time visual input from the user's surrounding, and then capture data is processed using machine learning algorithm to extract and simplify the essential visual features, and then processed visual features will translate it into the electrical stimulation patterns optimized for control interpretation, and then the encoded signals are delivered directly to the primary visual cortex, which is, as you can see, is on the occipital lobe, and then via the electrocorticography, which is the ECOG, and then the brain will receive and interpret the signal, enabling the user to perceive rudimentary forms of vision. And now you'll get.
So to be exact, we can treat 4 sorts of blindness. In a sense, they are all temporary blindness and partial blindness, which are caused by retinal damage, corneal damage, optical nerves, and visual cortex. According to our research by my friends, we can cure retinal damage by retinal neurostimulations and optical nerves damage by our gene therapies, and we can also cure the corneal damage by UV rays. So let me answer the 2 questions. Let me tell you about the 2 questions we were asked the most. Number 1, what makes you think that your product is gonna be cheap or like affordable for the Asians or like the whole populations? Normally, about we found a range for like how much people generally pay for such surgeries, daily checkups, and the medications. They pay a huge amount, and our product, it comes under the range which is 10 times or like almost 15 times cheaper than that. Also, we were asked what makes you think that like this is possible. We got this all checked up by the associate professor, Mr. Panamsak Mimun. So we thank him as well.
So here are the possible benefits that the OptiCortex provides. Firstly, the OptiCortex restores partial vision by directly stimulating the brain's visual cortex. Also, AI-powered real-time input detects and translates the surrounding environment into brain signals, enhancing spatial awareness. So I said by 30, you have to give it. I said by 30 seconds, you have to give it to me.
Thank you, Group 48, for a fantastic presentation. You did very well. And that concludes our final pitch. So why don't we have one more massive round of applause for all incredible teams for their dedication and creativity? The passion and ingenuity we've seen this afternoon are truly a testament to the future of science in Asia and beyond. To all the students, your dedication is inspiring. And I also would like to extend a heartfelt thank you to our judges and the award committee, who now have the very difficult task in their deliberations. According to our schedule, the award committee meeting will take place actually right after this until probably 5 o'clock. And the moment we all be waiting for, the closing ceremony, will commence at 5 o'clock, where the medals will be announced. So you don't have to wait too long. Thank you once again to everyone for being such a wonderful audience. And please enjoy the short break right after this. And we look forward to seeing all of you at the closing ceremony at 5 o'clock. Thank you. Goodbye for now.